The fertilization is taking place outside the reproductive tract of the female or outside the uterus of the female. It is called as in vitro fertilization. ZIFT means zygote intra-fallopian transfer and IT means intrauterine transfer. ICSI, intracytoplasmic sperm injection. Hello everyone, welcome to another session on reproductive health. I'm Dr. Divya, Biology Faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session, we had studied about the various sexually transmitted diseases, how to prevent these sexually transmitted diseases, and we had also studied about that these sexually transmitted diseases, they cause infertility in male as well as female. And what were the reasons for infertility in male and female? All this we had discussed. So in this session, we shall study about how a female can bear a child even if she is infertile because of various technological innovations that has taken place in today's world in medical fields. So these technologies which are related to reproduction or the female reproduction are called as assisted reproductive technologies. So these assisted reproductive technology can be adopted by those couples who are childless. So the couples, they could be assisted in order to have children through certain special techniques, and these techniques are commonly known as reproductive technologies or assisted reproductive technologies. They assist, they help the couple to bear a child in spite of them being infertile. So that is why it is called as assisted reproductive technologies. So under this assisted reproductive technologies, there are various methodologies that are involved. First one is IVF, which is called as in vitro fertilization. Then we have IUT, which is intrauterine fertilization. Then we have ZIFT, we have GIFT, then artificial insemination, we have ICSI. So all these in detail, how it is done, what are the procedures that are followed in these technologies, we'll study in the coming slides. So if all these technologies fail in spite of the couple taking up or going in for all these assisted reproductive technologies, and in spite of that, if they're not able to have a child, then there is always another option called as adoption. So adoption is wherein you adopt a child. So that adoption also, it should be legal. It should be done through the government. So it should be a legal adoption. So legal adoption is the best method for couples who are looking for to increase their family or looking for parenthood. So those couples who are not interested in adopting these assisted reproductive technologies or who have failed to bear a child in spite of undergoing treatments using these technologies, they can always go in for legal adoption. So we shall study about the different assisted reproductive technologies that are there in this today's world. So first one is in vitro fertilization. So in vitro fertilization. So in vitro fertilization, it is nothing but in vitro means outside the body. Whatever experiments is done, it is done outside the body in the laboratory condition. So for that, the term is used as in vitro. Here, fertilization is brought about in the laboratory condition outside the female reproductive system. That is why it is called as in vitro fertilization. So in vitro fertilization takes place outside the body of the female, wherein after that the embryo is being transferred. So that is called as ET, that is embryo transfer. So after in vitro fertilization, embryo will be transferred into the female reproductive system. So in this method, it is also known as the test tube baby. Why? Because here all the fertilization process is done either in a petri plate or in a test tube, and then that fertilized embryo that is formed, it is transferred into the female reproductive tract. So those children that are born out of this in vitro fertilization technique, they are called as test tube baby, and this particular method is called as test tube baby program. In this particular method, the ova, that is egg, which is there in the female. So the female is the donor because she will be giving the egg to the laboratory technician. So the egg or the ova will be collected from the female and the sperm will be collected from the male. So from the husband, here both the husband and the wife or the couple, they play a role of donating. That is why they are called as the donor. So the female donor will donate her egg or will give the egg to the laboratory, and the male donor will give the sperm to the laboratory. So in the laboratory, they will collect those two and they will fertilize them or they will fuse them, and then the zygote that is formed, after fusing, the zygote that is formed, it will be transferred into the uterus of the female. So you can see here the egg, it is collected, the egg or the ova, it is collected from the female. This is put in a petri dish. So either in a test tube or in a petri dish it is put, and so this is the female donor. So this is the female donor, and from the male donor, the sperms. So these are the sperms. Sperms will be collected. So all the sperms will not be used. Here also they will check for the motility of the sperms and they will check for the fertility of the sperms. And those sperms which are capable of fertilizing, only those will be transferred to the petri plate. So now in this petri plate, you have the female egg and you have the sperm, male sperm. So this will be fused together and the resulting zygote that will be formed. So this is the zygote. So the zygote that will be formed, it will be put into the uterus of the female, and there the child will develop. So since the fertilization is taking place outside the reproductive tract of the female or outside the uterus of the female, it is called as in vitro fertilization. And after this, because the embryo transfer is done, there is one more process called as ET, which is nothing but the embryo transfer. So this is how in vitro fertilization occurs. So next, moving on to in IVF or in vivo fertilization. So IVF can stand for in vitro fertilization and it can also stand for in vivo fertilization. So in vivo means inside the body of the human being. Here the zygote will not be formed outside the body of the female, but only the sperm will be transported. So we'll study about that. So what is done here is in vivo fertilization, the embryos are formed by the fusion of the gametes within the female. So what is the gamete here? It is nothing but the sperm. So the sperm is made to fuse with the gamete. But where will the fusion take place? It will take place inside the female uterus. But in vitro fertilization, where did the fusion take place? The fusion of the female ova or the egg with the sperm was done outside the body of the female, that is outside the uterus in a petri dish. That is why it was called in vitro fertilization. But in the case of in vivo fertilization, the gametes will fuse with the egg that is present inside the female itself. The egg will not be taken out. It will be there inside the female itself, and to that particular egg, the fertilization will be done. Therefore, it is called as in vivo fertilization. And this in vivo fertilization, it mainly helps those females who are not able to conceive. So say for example, the female will be bearing a good productive egg or ova, but the male sperm will not be able to properly travel towards the female egg. So therefore, to assist the travel, the physician or the laboratory technician, whoever is there, they themselves will insert the sperm into the female reproductive tract. So you can see here what they do is they first collect the sperm. Here only they'll collect the sperm. They will not collect the ova or the egg of the female. They'll only collect the sperm, and then they will sperms which are motile. Only those sperms which are motile will be selected, and or those sperms which are capable of fertilizing, only those will be selected, and through a catheter it will be directly introduced. So once the sperms are introduced, so you can see here the sperms are introduced, those sperms which are highly motile, they will travel, and whatever egg or the mature egg or the follicle that is present, that will be fertilized. So you can see here they will start to move. Only those sperms that are fast in motility, they will move and they will fertilize the egg. So this is how the fertilization occurs here. Fertilization is occurring inside the body. In the case of in vitro fertilization, fertilization was done outside and then the zygote was put into the female uterus. But here fertilization will be done inside. Only the sperm will be put inside the female uterus, or wherein the sperm will travel and it will fertilize the egg and the zygote will be formed, resulting in the embryo development and the fetal formation. So this is how it will help. So next there is one more technology, that is ZIFT and IT. So ZIFT means zygote intra-fallopian transfer and IT means intrauterine transfer. So ZIFT is nothing but zygote intra-fallopian transfer. You are putting the zygote into the fallopian tube. You are transferring it there. That is why it is called as zygote intra-fallopian transfer. And one more is intrauterine transfer. You will be transferring it to the uterus. It will be transferred to the uterus. That is ZIFT is nothing but zygote intra-fallopian transfer, and IT is nothing but intrauterine transfer. So here in the case of ZIFT technology, when the zygote or the early embryo, they develop into the 8 blastomere stage, it will be transferred to the fallopian tube. Therefore, it is called as ZIFT. So, from where will the 8 blastomeres be developed? Again, here for that, they will be collecting the egg of the female. They will be collecting the egg and they will be collecting the sperm. So, here the sperm and the egg, they will allow both of these to fuse, and once they become the 8 blastomere stage, it will be introduced. So the 8 blastomere stage. After fertilization, in vitro fertilization, what was done? Soon after the fertilization occurred, it was transferred to the uterus. But in the case of ZIFT, what is done is the sperm will be collected, the egg of the ova of the female will be collected, and they will be kept for a certain time until it reaches the 8 blastomere stage. And once it reaches the 8 blastomere stage, this will be collected with the catheter and it will be introduced into the fallopian tube, and this it will travel. So here inside the fallopian tube you will have this 8 blastomere stage being inserted, and this will travel and it will incorporate itself in the uterus. So this is how it occurs in the case of ZIFT. And when it comes to intrauterine transfer, the zygote or the embryo, once it reaches more than 8 blastomere stage, it is directly placed in the uterus. I hope now you can understand the difference between the two. In the case of ZIFT, once the zygote or the embryo, after the fusion or fertilization of the sperm with the ova, it will be introduced into the fallopian tube. From the fallopian tube, it will travel to the uterus and settle there. But in the case of intrauterine transfer, again, this zygote, again it should become maybe 16 blastomere stage or more than that. Once it becomes more than the 8 blastomere stage, it will be inserted to the uterus. So in ZIFT, the 8 blastomere stage embryo was inserted into the fallopian tube. In uterine transfer, the 8 blastomere stage embryo, the embryo which is having more than 8 blastomeres, they will be directly inserted into the uterus. Here fallopian tube is not at all involved. It will be directly inserted to the uterus using a catheter. So that is nothing but intrauterine transfer. So next we have one more technology, that is assisted reproductive technology, which is called as GIFT. GIFT, which is gamete intra-fallopian transfer. So in this method, the ovum, the ova or the egg from the female will be collected from the donor and it will be transported into the fallopian tube. And also in this particular gift, if the female, because of some reason she is infertile and she is not able to produce the ova or the egg, she can always go in for a donor's egg. So some other female, she can use the egg of a donor female and that this particular procedure can be done. So what they will do is they will collect the egg or the ova from the female and it will be transferred into the fallopian tube of another female who cannot produce. So what happens here is, because of some reason, the uterus of the female will be intact. No problem would have occurred in the uterus, but some problem would have occurred in the infertility, that is in the fertility of the female, wherein she will not be able to produce the egg. So in such cases, when the uterus is fine but in spite of that she is not able to produce the egg or the ova, from a donor or from any other female, the compatible eggs will be collected and then that egg will be transported or placed into the fallopian tube or transferred into the fallopian tube of the female who is planning to conceive. So this is the best technique. So the uterus will be of the female who is planning to conceive, but the egg will be from some other female, because that particular female who is planning to conceive or get pregnant, she will not be capable of producing the egg. So that is about gamete fallopian transfer. So what is done is, here again this from a donor female, from any other donor female, the egg will be collected and the sperm will be collected, and once it reaches the blastomere stage, it will be directly placed in the uterus. So here the only difference, or it will also be placed in the fallopian tube. So from the fallopian tube it will travel and it will implant itself here. So this is the difference between that is in GIFT, this will be done. Again, in the case of in vitro fertilization, what was done? It was directly, as soon after fertilization occurred, the embryo was directly transported into the uterus. But in this case, it will be transported to the fallopian tube. So this is how it actually occurs. Next is intracytoplasmic sperm injection, that is ICSI, intracytoplasmic sperm injection. So it is something related to the cytoplasm of the ova or the egg. So that is why it is called as intracytoplasmic sperm injection. So in this method, a single sperm will be collected and it will be directly injected into the ova using a micro needle. So this method is usually used in cases where the male are infertile. So if the male is not capable of producing sperm, or if they have a low sperm count or zero sperm count, or even though if they produce the sperm, the majority of the sperms will not be motile. In such conditions, they are called as infertile. So when the male is infertile, they can use the sperm from a donor male. Whoever is capable of donating, the sperm will be used from the donor male, and only one sperm will be selected. Only one sperm, that is a good quality sperm, will be selected, and that good quality sperm, with the help of the micro needle, so this is the micro needle. So with the help of the micro needle, it will be directly injected into the ova or the egg of the female. Ova or the egg, it will be directly injected. So this whole procedure will take place inside the female reproductive system. So you can see the sperm here. It will be directly, only one sperm will be directly injected. So directly to the ova region, that is in the ovary where the mature ova is being present, there itself the sperm using a microneedle, it will be injected to that particular ova. So when the sperm is injected, it will fertilize the ova and the female can bear a child. So this method is usually followed in cases where the female has a good reproductive system or reproductive health, but the male is infertile. In such condition, this particular intracytoplasmic sperm injection is done. It is called sperm injection because one single sperm is being injected into the ova or the egg of the female. Next, we have one more important technology for helping the female bear a child. It is called as artificial insemination. So in this technique, the semen will be collected or the sperm will be collected from the husband or a healthy donor. So if the male is infertile and is not capable of producing sperm, then they can also collect a sperm from another male who is a healthy donor. And here it will be artificially introduced either into the vagina or to the uterus. So it is also called as IUI, that is intrauterine insemination. So artificial insemination is also called as intrauterine insemination because the insertion or the introduction of the sperm can be done into the uterus as well. So this method is used in infertility cases wherein again here if the male partner, if he is infertile, then this procedure can be used. So what is done here? Again here, not just the male, even if the female is also infertile, if both the male and the female in a couple is infertile, then this procedure can be done. So you have the donor. So this is the donor sperm. So the donor sperm, he'll donate the sperm and that particular sperm will be injected into the uterus. It will be directly injected into the uterus and this sperm, whichever is capable, will go and it will fertilize the egg and next the zygote will be formed. So that is how it is done here. So this is nothing but artificially you're inseminating. You're collecting the sperm from someone else or a capable donor and then then the sperm is being inserted through an injection. It is being inserted into the female. So that is nothing but artificial insemination. So there are certain disadvantages when it comes to assisted reproductive technology. So the main advantage is that those couples who are not capable of having a child can go in for assisted reproductive technology. So in spite of this having a wide range of advantage to the couple, it also has its own disadvantages. So the first one is it requires extremely high precision handling because all this is done in the laboratory condition. It is we are going against the nature. We are doing it in the laboratory condition. So therefore, you cannot just rely on somebody to do this. There should be a specialized center or IVF centers or fertility centers wherein they should have very sterile or clean laboratory setup. So in that laboratory setups, all this should be done especially by specialized professionals. All these will not be done by the gynecologist. There will be people or laboratory technicians who are specially trained for doing all this insemination experiments and all that. So it requires extremely high precision handling by specialized professionals and expensive instrumentation. So not all the people or all the couple can afford this assisted reproductive technology because IVF and all that is very, very costly. So they will not be able to afford. So that is one of the major disadvantage of assisted reproductive technology. And therefore, because it is very expensive, it is limited only to a number of people. Why? Because assisted reproductive technologies, when the couple goes in for all these different technologies of that is assisted reproductive technologies, they'll have to go in for clinical visits, regular checkups, quite often when compared to the natural method of conception. So in such cases, in places where there is no availability of these regular visits by these technicians and all that, it is very difficult for the people to follow these assisted reproductive technology. So therefore it is only affordable to only a limited number of the people or the population. One of the major reason why it is not affordable by all is because it is very expensive. So therefore all the people cannot, or all the couples cannot go in for assisted reproductive technology. One more is, it is emotional, religious and social factors also play a major role, because usually how much ever the people in a society are educated, still they will say that the couple is not able to, they look upon them as something, as if they have done a sin or something. They will say you have done a mistake, you're not capable of bearing a child. They'll start blaming the female. So, and also they will say you should not go in, you should not go against the nature, you should not go in for taking up all this reproductive technology methods such as IVF, be it ZIFT, GIFT and all that. So also emotional, and they will also say religious. They will say the God should give you a child. So don't go visit the hospitals, don't go for all these artificial technologies. So therefore emotional, religious and also various social factors are one of the reasons why the people, if they are not able to have a child, they like hinder themselves, or they take a step back to go in and get this assisted reproductive technology procedures done. And also sometimes if because of all these disbeliefs, if or in spite of going in for assisted reproductive technologies, even then if the couple, they are not able to bear a child, then they can always adopt. So adoption also, it is one of the biggest factors. The couple should be ready. They should be ready both emotionally, religious and socially. They have to be ready to adopt, because even then people will start blaming, saying that maybe you have adopted a baby, we don't know which religion it belongs to, what social status of that baby is. So before going in for adoption, all that should be taken care of about it. So whenever we can go in for an adoption, it is better to go into legally, that is through a government organization or through the norms, following the norms of the government. It is better to go in for adoption. So emotional, religious and social factors are also deterrents when it comes to adopting methods. So different adopting methods, be it adopting is also, be it going in for assisted reproductive technology, or be it adopting a child from somewhere else. So this was about assisted reproductive technology. So what you have learned in this particular session, just don't limit it for the study purpose or for the sake of writing exams. All this can be implemented in your life. Not just that, because the knowledge that you have gained from the reproductive health, you can also educate other people who do not get a chance to learn about all these. You can always educate them, and you can always bring a social awareness with the people who are surrounding you. So if something illegal or something bad is taking place in the society related to the reproductive issues, you can always, don't be shy to put a step forward and please start educating people, because since you have learnt about all this, now you can start educating people about all these things. Don't always shy away from all these things. So I hope you have understood this complete session of reproductive health very well. So in the next chapters and the coming sessions, we shall meet again. Thank you.